Question number 1. What is the process in which one strain of bacteria is changed by the genes of another strain of bacteria? The answer is transformation. When I think of transformation, I think of transformers. So you have a small piece of bacterial DNA, and that small piece gets incorporated into another bacteria, like Optimus Prime. And then all of a sudden, you get a new transformed bacteria that not only has its own original DNA, plus it has the DNA of the small piece that got incorporated into it. All right, question number 2. Who is the scientist responsible for discovering the process of transformation? The answer is Frederick Griffith. So Frederick Griffith discovered that genetic material from bacteria can be incorporated into a completely different bacterial cell. And let's see, how can we remember Frederick Griffith? Because we're going to go over a lot of scientists. All right, when I think of Frederick Griffith, I think of Freddy Krueger, or should I say Freddy Krueger. So Freddy Krueger, what do we know about Freddy Krueger? We know that Freddy Krueger killed things. Well, killed people, right? Well, Frederick Griffith killed mice. So Frederick Griffith discovered the process of transformation by experimenting on mice. Okay, remember that. Question 3. Who is a scientist who mixed heat-killed pneumonia-causing bacteria with harmless bacteria and injected the mixture into mice? We know that guy. That's the killer guy. It's not Freddy Krueger, it's Freddy Krueger, so it's Frederick Griffith. So when I think of Frederick Griffith, I'm going to think of Freddy Krueger, or shall I say Freddy Krueger, and I'm going to picture his claws as syringes. That'll help me remember that he's the guy that injected pneumonia into mice, and these mice that caught pneumonia died. Question 4. In Griffith's experiment, what happened to the mice that got the mixture of harmless bacteria and he killed pneumonia-causing bacteria? Well, they died of pneumonia. So when he mixed the pneumonia-causing bacteria that was killed into the harmless bacteria, it got transformed. And all of a sudden, that harmless bacteria was not so harmless anymore, and the mice died. Question 5. Who led a group of scientists to discover that DNA stores and transmits genetic information? Well, that person is Oswald Avery. So remember, Frederick Griffith discovered the process of transformation. Oswald Avery, and when I think of Avery, I think of aviator. So I'm going to picture Ozzy the aviator here. So Oswald Avery took Griffith's experiment one step further. He did the same experiment that Freddy Griffith did, but he also figured out that it was DNA that was the genetic material. They didn't know what the genetic material was. Well, Avery figured it out. Ozzy the aviator. Question 6. Who repeated Griffith's experiment and took it one step further to identify DNA as a genetic material? All right, well, first came Freddy Krueger, or Frederick Griffith. Then it's Ozzy the aviator. So Oswald Avery. So Oswald Avery did the exact same experiment that Frederick Griffith did. All right, except he took it one step further and actually identified DNA as genetic material. So when you think of Ozzy the aviator, well, maybe you should put those syringe claws in his hands because he did the same experiment as our good buddy Freddy Krueger or Frederick Griffith did. He killed mice, but he took it one step further, found out that DNA was a genetic material. Question 7. Were scientists 100% convinced by Avery's work that DNA was the genetic material? No, they wanted better evidence. Many scientists didn't believe that Avery provided conclusive evidence that DNA was indeed the genetic material. Well, we have a name for people like this where I'm from. Question 8. Who conducted an experiment with bacteriophages and radioactive isotopes? Alfred Hershey and Martha Chase. So the team got a bacteriophage, which looks like this spider-looking thing right here, which is basically a virus that infects bacteria, and made it radioactive. They made the protein head radioactive, they made the DNA in the inside radioactive, and they found out that it was the DNA, not the protein, that got incorporated into the bacteria that got infected. Therefore, DNA was a genetic material. And with a little imagination, that Hershey kiss looks a little bit like the head of the bacterial phage. So there you go. And if you can commit that image to memory, that'll help you remember that Hershey and Chase did the experiment with bacteriophages and radioactive isotopes. Question number 9. What did Hershey and Chase determine? Well, they determined that DNA was a genetic material. Question number 10. Whose experiment confirmed Avery's findings that DNA is the genetic material? The answer is Hershey and Chase. Remember, Avery already found that DNA was a genetic material, but scientists weren't 100% convinced. So Hershey and Chase come along and they use bacteriophages and radioactive isotopes to conclusively confirm that DNA was indeed the genetic material. Question 11. What are building blocks of nucleic acids? The answer, nucleotides. Basically, nucleic acids are a bunch of nucleotides put together. And remember, DNA is a nucleic acid. The other nucleic acid's RNA. Question 12. What type of macromolecule is DNA? The answer, nucleic acids. Remember, in biology, there are 4 main types of macromolecules: nucleic acids, proteins, lipids, and carbohydrates. DNA is a nucleic acid, and the other one is RNA. RNA is also a nucleic acid. Question 13. What is DNA made of? Well, it's made of a phosphate backbone, a 5-carbon sugar, specifically deoxyribose, and a nitrogenous base. Question 14. Which nitrogenous bases make up DNA? Well, those are adenine, guanine, cytosine, and thymine. Question 15. What kind of bond connects adjacent nucleotides in DNA? The answer is covalent bonds. Remember, DNA is made up of building blocks, and these building blocks are nucleotides, and each nucleotide is made up of a phosphate backbone, a 5-carbon sugar, and a nitrogenous base. Well, each one of those building blocks is connected to the next one through a covalent bond. Question 16. What type of bond connects the nitrogenous bases across from each other in DNA? The answer is hydrogen bonds. Remember, DNA is double-stranded, and so you have a string of nucleotides on one strand, and you have a string of nucleotides on another strand, and the nucleotides that are across from each other are connected by weak hydrogen bonds. Question 17. Nitrogenous bases fall into 2 general categories: double ring structures versus single ring structures. What is the name of the single ring category, and which bases belong to this category? The answer is pyrimidines, and cytosine and thymine belong to the pyrimidine category. So there are 2 types of nitrogenous bases. You have the ones with double rings, and you have the ones with a single ring. This question is about the one with a single ring. And when I think of a single ring, I think of the base of a pyramid. The base of a pyramid is definitely not made up of 2 rings, right? So when you think of the base of a pyramid, think of 1 ring. Okay, now think of somebody named TC. And TC lives inside this pyramid. So if you can remember TC living inside of a pyramid, and T stands for thymine, C stands for cytosine, TC belongs to the single ring or pyrimidine group. Question 18. What is Sharkoff's rule? Well, Sharkoff came up with the base pairing rules. He discovered that adenine and thymine are always in the same ratio, and guanine and cytosine are always found in the same ratio. So he didn't know why that was, but he knew that A went with T and that C went with G. And it's super convenient that his name is Sharkoff because he's got a C in his name and a G in his name and an A in his name, and, well, I just stuck a T over there. So Sharkoff rule. And if you're having trouble remembering which letters go with which, well, the letter C kind of looks like the letter G, right? You can modify the C into a G. So that'll help us remember that C goes with G, and then from there you can remember that A goes with T. Question 19. Who used X-ray crystallography to get information about the structure of DNA? The answer is Rosalind Franklin. It was actually this photograph known as Photo 51, which was taken by, I believe, one of Franklin's PhD students, that allowed Watson and Crick, those are the 2 people that are credited for coming up with the structure of DNA. Well, it was her work that allowed them to figure out that DNA was a double helix. So essentially, Her work was the missing link in the discovery that DNA was indeed a double helix. Number 20. Did Rosalind Franklin discover the structure of DNA? The answer is no, but her work greatly contributed. Without her work, it would have been much more difficult for Watson and Crick to figure out that DNA was a double helix. Question 21. Who was originally credited for discovering the structure of DNA? The answer is James Watson and Francis Crick. They came up with a double helical model, but they drew upon work of other people, such as Sharkoff with his base pairing rules, and also Franklin's X-ray crystallography, where they used the data from her work to figure out that DNA was a double helix. And if you need help remembering that James Watson and Francis Crick were the ones that actually came up with the structure of DNA, but they drew upon other people's ideas, well, think of a light bulb. A light bulb has watts. And a light bulb also means ideas. So it was Watson and Crick who drew from the ideas of other people, such as Franklin, and came up with their model. All right, question 22. Whose work allowed Watson and Crick to realize that DNA was a double helix? The answer is Rosalind Franklin. Remember, it was Rosalind Franklin's famous photo 51 that allowed them to realize that DNA was a double helix. Question 23. Arrange the following people chronologically according to their scientific contributions. All right, first we talked about Freddy Krueger, or Frederick Griffith, and then we talked about Avery, who also has claws just like Griffith because they both killed mice. So Frederick Griffith discovered transformation. Avery took his transformation experiment a step further and found out that DNA was a genetic material. Then we have Hershey and Chase that actually confirmed Avery's work that DNA was a genetic material. Then we had Watson, Crick, and Franklin who actually contributed to the structure of DNA. Watson and Crick are credited for the structure of DNA, but they couldn't have done it without Franklin's work. Question 24. Do the two strands of DNA run in the same direction, or are they anti-parallel? The answer is anti-parallel. So when I think of DNA, I think of shoes in a shoebox. Just like shoes have heels and toes, DNA has a 3' end and a 5' end. And if you have the 3' end on one side, then right across from it, you're going to have the 5' end on the other side. So when I think of DNA, I think of shoes in a shoebox, and DNA is anti-parallel. Question 25. What holds the two DNA strands together? The answer is weak hydrogen bonds. A lot of times textbooks draw hydrogen bonds as little red dots, and that's what holds those two adjacent strands together. Question 26. Are hydrogen bonds strong or weak? They are actually weak bonds, and they need to be weak bonds because during DNA replication, they get broken apart. Question 27. Why is DNA held together by weak bonds? The answer is so it can be easily unzipped for replication. And notice by the lack of red dots that when DNA gets unzipped, those hydrogen bonds are broken. Question 28. The fit between AT and CG. So what's the fit between adenine and thymine and cytosine and guanine? That's known as complementary base pairing. So, and remember, Sharkoff figured that out, that adenine goes with thymine and that guanine goes with cytosine. Question 29. Whose model explains Sharkoff's rule? The answer is Watson and Crick. So Sharkoff figured out that A is always in the same proportion as T, and C was always in the same proportion as G, and Watson and Crick figured out why that was so. Question 30. The cell makes a copy of itself during the cell cycle. During this process, the nucleus divides during mitosis. At what stage of the cell cycle involves DNA replication? So really the question is asking, at what stage of the cell cycle involves DNA replication? And the answer is the S phase. So the cell cycle has two main components, interphase and the mitotic phase, where mitosis happens. And DNA replication occurs during interphase, and more specifically, the S phase part of interphase. Question 31. What is the process of copying DNA? The answer is DNA replication. That's when one molecule of DNA replicates into 2 molecules of DNA. Question 32. During DNA replication, each strand of the double helix serves as a blank for the new strand. The answer is a template. So each strand on the DNA molecule is basically a guide for the copied strand. So DNA gets unzipped and both portions that are pulled apart become a guide for the new one to be copied onto. So each template strand comes from the original DNA molecule and a new strand is copied onto that. Question 33. What enzyme is responsible for unzipping DNA during DNA replication? The answer is helicase. Anything that ends in ase is an enzyme. And when I think of DNA helicase, I think of a helicopter. And I think of the blades of a helicopter busting up those or breaking apart those weak hydrogen bonds. So when I think of helicase, I think of helicopter, and I think of a helicopter blade busting up hydrogen bonds, allowing DNA to be unzipped. Question 34. During DNA replication, the 2 strands are unzipped. Where exactly on the DNA molecule does replication occur once the molecule has been unzipped? And the answer is at the replication forks. So the replication fork is the area where the 2 strands branch apart, and that's where DNA replication begins. Question 35. During DNA replication, if the base on the old strand is adenine, what base is added to the new strand? All right, and the answer is thymine. And the way you can remember that is, well, we know that C goes with G because C and G look the same. Therefore, A goes with T. So what's opposite adenine? Thymine. Question 36. During DNA replication, a strand that has the base sequence TACGTT will produce a strand with the base sequence blank. The answer is ATGCAA. And well, we know that A goes with T and C goes with G. So across from every T, you have an A. Across from every A, you have a T. Across from every C, you got a G. And across from every G, you have a C. Question 37. Nitrogenous bases fall into 2 general categories: double ring structures versus single ring structures. What is the name of the double ring category? Which bases belong to this category? So remember, TC lives in a pyramid, and pyramids are made out of a 1-ring base. So I know that thymine and cytosine belong to pyrimidines, which means that the other ones belong to purines. All right, so the answer is purines, and adenine and guanine belong to purines. Question number 38. Does DNA replication result in the original DNA molecule and then a completely new DNA molecule? Basically, the question is asking, do you get a baby DNA molecule from a parent DNA molecule? And the answer is no. Each strand will have 1 new strand and 1 original strand. So when DNA gets unzipped, it never actually comes back together again. Each strand becomes half of a new DNA molecule, and the other half is copied onto that template strand. Question 39. What is the main enzyme involved in DNA replication? Well, the answer is DNA polymerase. When I think of DNA polymerase, I think of a Pac-Man with glasses. And instead of eating up little bits, Pac-Man is putting them down. So notice what's coming out of Pac-Man's mouth is the same color as the copied strand. Also notice that Pac-Man has glasses on, because DNA polymerase does 2 things. It reads the DNA and also copies the complementary nucleotide onto the template strand. And remember, a nucleotide has a 5-carbon sugar, a phosphate backbone, and a nitrogenous base. So DNA polymerase has to actually read which base is already there. So for example, if there's an adenine there, it's going to put down a thymine. And if there's a thymine there, it's going to put down an adenine. If there's cytosine there, it's going to put down a guanine. Question 40. What kind of molecule is DNA polymerase? Well, DNA polymerase ends in ACE, and anything that ends in ACE is an enzyme. Question 41. What is the name of the enzyme that lays down complementary base pairs during DNA replication? Well, that's those little Pac-Man guys. That's DNA polymerase. Question 42. What happens if DNA polymerase accidentally puts down the wrong base during DNA replication? Well, DNA polymerase proofreads. It will remove the wrong base and insert the correct one, which is why when I think of DNA polymerase, I think of Pac-Man with glasses, because DNA polymerase is not only just spitting down new nucleotides, it's proofreading. It's making sure that if it accidentally put a C with an A, it kicks out that C and puts a T. Okay, now the multiple choice portion of your exam. Question 43. The process by which one strain of bacteria DNA is changed into another strain is called A, duplication, B, transcription, C, transformation, or D, replication. And the answer is transformation. And that's what that guy Griffith figured out, Frederick Griffith. Okay, question 44. Bacteriophages are A, enzymes, B, viruses, C, sections of DNA, D, types of bacteria. The answer is viruses. And remember, that bacteriophage looks like a spider. The head of the spider looks a little bit like a Hershey kiss. I'm just saying. So Hershey and Chase are actually people that worked with bacteriophages. Okay, question 45. A nucleotide does not contain A, a phosphate group, B, a 5-carbon sugar, C, an amino acid, D, a nitrogenous base. And the answer is amino acids. Amino acids are something that proteins are made up of, not nucleotides. Question 46. According to Sharkoff's rule of base pairing, which of the following is correct about DNA? Well, I remember the letter C looks like the letter G, therefore C goes with G, and A must go with T. All right, so the answer is D. A goes with T, and C goes with G. Question 47. The bonds that hold the two strands of DNA together are a result of A, phosphate groups attracted to each other, B, strong bonds between nitrogenous bases and sugar phosphate groups, C, weak hydrogen bonds between nitrogenous bases, and D, carbon bonding. The answer is C, weak hydrogen bonds between nitrogenous bases. Remember, those weak bonds get unzipped, and helicase helicopter unzips those weak hydrogen bonds. Okay, question 48. In eukaryotic cells, almost all the DNA is found in the A, histone, B, endoplasmic reticulum, C, nucleus, D, cytoplasm. The answer is C, nucleus. So in a eukaryotic cell, which is basically a non-bacterial cell, the DNA is going to be found in the nucleus. Question 49. In prokaryotic cells, almost all the DNA is found in the A, histone, B, endoplasmic reticulum, C, nucleus, D, cytoplasm. And the answer is D, cytoplasm. So prokaryotic cells are bacterial cells, and they don't have a nucleus. So their DNA is found in the cytoplasm. Whereas eukaryotic cells, it's found in the nucleus. Question 50. The primary enzyme involved in connecting individual nucleotides into a DNA molecule is A, histase, B, DNA polymerase, C, ribose, D, helicase. I know it's not ribose because that doesn't end in ase. Enzymes end in ase. All right, so we can eliminate ribose. And the answer is actually going to be B, DNA polymerase, because remember, DNA polymerase is little Pac-Man guys, and they not only lay down nucleotides, they connect adjacent ones to each other. Question 51. The Y-shaped region that results when the two strands of DNA separate is called A, replication fork, B, complementary strand, C, template, D, base pairing strand. And the answer is A, replication fork. Remember, the replication fork is what occurs when the two strands separate from each other when the helicase breaks them apart. All right, guys, that's the last question on the test. If you like this video, please hit the like button and subscribe if you want to see more videos like this, and I'll see you next time.